Hello, everybody, and welcome to The Crucible, our weekly discussion focusing on the scientific topics we find important as secular humanists. I'm your host, Skids the Clown, and before we get started, I'd like to say hi to everybody joining us this evening. Thanks for stopping by, and if you like what you see on this channel, please remember to hit that like button, share the video, subscribe to the channel, and if you'd like to see even more content from the Promethean Secular Frontier, make sure you check out our other shows on this channel, which includes the Secular Soapbox, The Bible Says What, and the Borg Skeptic Show. And don't forget to visit our website where you can find links to all of our content, including our Discord channel, which has lots of great stuff. Now, tonight we're going to be discussing abiogenesis. And here to help me bring life to this topic, get it? Life to the topic. Let me bring in the rest of our panel, starting with our other host for the evening, the Borg Skeptic.
Hello, how are you doing tonight?
I'm doing fantastic. How are you doing?
I'm doing absolutely fantastic myself, and before we get into anything, I'd just like to say thank you to all of the subscribers and all of our supporters, because we finally hit our first milestone, 100 subs. I know it doesn't sound like a lot, but that's the first major hurdle that we've passed, and yeah, just thank you to everyone who's helped out with that. Tonight's topic is abiogenesis, like you mentioned. Mm-hmm. And I would like to introduce our guest for tonight, which is Dragnot. We all know him. We've all seen him around. He's a big name out there in the YouTube community, and he's been doing some great things. He has a bachelor's in science in biology, with a specialization in cellular, molecular, and biotechnology. He's currently seeking a master's in science in molecular biotechnology, and he's doing a thesis in the field of biochemistry researching cell aging. So let's bring him on the screen to get his expert opinion on abiogenesis. How are you doing tonight, Dragnot?
I'm doing all right. I'd hardly call myself an expert, but no, I appreciate it.
I say you're more of an expert than myself, simply because I only understood half the words in your introduction. So you must be doing something right.
Yeah, just coursework. But no, it took a bit for me to finally get my bachelor's, but it takes a lot. Like for any topic or any niche topic, I should say, having the requisite knowledge will take some time and understanding. But there's a way that we can sort of break it down so that everyone can have at least a firm understanding of what currently is going on.
Awesome. So to get started tonight, why don't you tell us and our audience a little bit about yourself and kind of what your interest in secular humanism is?
If you may have seen me on AXP or on the line, the Hang Up, I was pretty active back during the Google Plus days, and sort of having online discussions with Dias. And that's sort of taken a backseat to my education now. But I'd like to try and use my abilities now to help people who want to engage in that sort of thing more actively. And my contributions towards the secular humanism is to help prepare people to engage in these conversations, because we're in the midst of a global pandemic, and we're seeing that people really don't care about others, especially people who are immunocompromised. Right. Yeah. So this is the perfect opportunity to show folks, hey, the folks who are trying to make sure that in Austin, hey, the homeless are taken care of, secular humanists. People who want to make sure that that the immunocompromised are protected, secular humanists. And so it gives us a lot more visibility in this time of need.
Absolutely. So on topic tonight, can you tell us a little bit, and you might have to keep it down to a pretty high level on your description, what abiogenesis is?
All right. So when we think about abiogenesis, we sort of have to go back before there was any life, right? We hear the reductionist argument, oh, we're just bags of chemicals and stuff like that. Well, at some point in time, there were just the raw chemicals. There weren't any amino acids. There were just carbon. There was just oxygen. There's just hydrogen. Yes, some of them combined into water, but we did not have this thing that we call life. We have polymers, right, created from some of these same elements that don't have the emergent properties that we see in life. And when I say emergent properties, it's when you have a substance combined with another substance, and it takes on new characteristics that's not seen in either of those other two substances alone. So this is the prevailing idea that at some point, something had to combine under some sort of conditions in order to create the life that we see now today.
And what kind of conditions make this possible? Is this something that we actually can see today occurring? Because it seems like it's something that could only be replicated in a lab and not through nature.
Right. So the way that we investigate this is that we have to sort of create a sterile environment, because there's no place on this planet that you're not going to find life. There are places in the deepest, darkest of depths that life still lives. And I know that's a little redundant, but you have what are known as extremophiles, where they thrive in these environments that would kill us. So you have to sort of create a sterile environment and then say, okay, what do we know about the conditions of early Earth? There are geological samples that are taken. There are atmospheric samples that are taken from those geological samples. And so we sort of surmise what the conditions were that would help kind of kickstart life. Because once the ball starts rolling, you know what, it's just going to keep going, because what doesn't work is going to get discarded, and the stuff that does work is going to stay and create what we have now. So we have sort of an idea of what may or may not have happened, but we've yet to sort of just create life de novo, just out of nothing, because there's so many constraints that we have to contend with, and there's also different ways. And and here's the thing: there may have been more than one way, right? Life was created, and so we can only investigate one at a time in order to sort of minimize all the variables. And that's sort of like where a lot of literature is at right now, is saying, hey, we think that one of these possibilities is that there was a very, very early precursor to our genetic material, and that whatever this substance is had the ability to catalyze its own reactions. And it's when you talk about a catalyst, it's something that helps a substance, in this case, let's say RNA, right? That RNA can catalyze its own reaction to maybe add or cleave off a section of itself, because these sort of chemical interactions take a certain amount of energy, and in order to acquire that energy, now we have proteins, enzymes that will assist in that. But if those enzymes aren't around, that's a huge hurdle for just raw materials to overcome. So there's our problem.
Yeah. So we're talking about, you just mentioned RNA there. Could you explain a little bit, just for the viewers and the lay people out there, the difference between proteins, RNA, DNA? Just to clear that up a little bit.
Right. So you can think of, as far as we go, RNA takes a few different forms. It is genetic material, just like DNA, except RNA is single-stranded. It's ribonucleic acid as opposed to deoxyribonucleic acid. It's very, very similar, except, like I said, DNA is double-stranded, RNA is single-stranded. You will have some cases of double-stranded RNA. There are viruses that exist that are double-stranded RNA, but RNA is sort of the precursor to creating DNA. And so far as our bodies go, we have RNA that's created out of, adenosine, thymine, cytosine, and guanine, while you have some of those same materials used for RNA, except you have uracil substituting out one of those nucleotides. So that being said, you have to sort of understand there's the recipe, that's the RNA. There are enzymes, which are proteins that are created not out of the materials that I said before, but those are created out of amino acids, a completely separate substance that assist in their job. So, and I know this is a lot to take in. There's a lot to take in. Amino acids go to proteins. You have these nitrogenous bases go to the genetic material, the RNA and the DNA. And so they all act in concert. The RNA acts as, well, in this example, the recipe, the enzyme, which is a protein that's made from amino acid, assists in reading that and turning that into yet another protein. Does that make sense?
Yeah, so it would be the enzymes are basically if you're talking about a recipe, they're basically the spoon which is mixing the pot.
You can say, yeah. That's a good example. Yeah.
So once we basically have this recipe, the enzymes, proteins, and everything like that, and something triggers to go from basically just the recipe to a simple life form, where does that go from there?
Right. So that's the hard part. We're not really sure. Right. So if you say that, hey, I can get some because we can artificially create RNA. We can artificially create some proteins even. Now, getting them to fold properly, that's a tough part, but it's possible. So getting them to act in concert to create life, we're really not too sure, because if we're talking about just one cell, you have a phospholipid bilayer, which acts as, you could say, like a plastic wrap that holds in all the goo, all the good stuff. And so with that, you have to sort of relegate different spots inside of that for different purposes. And trying to create those separately is a monumentous task. And that's why you have certain younger creationists go, well, obviously, since we can't do this, we can't figure out how all this acted, how all this was done at the same time, then obviously a deity had to have done it. When there's no requirement at all that they were created all together, and they could have just been very well created separately through natural processes. So, and that's a sort of bit of outside of my area, because I only really deal with cell aging, and the origin at which like some of these things came about is sort of still an open mystery, but it is being explored now in the literature.
And I definitely want to touch on the theistic creationism point of view on that, because I've heard the argument, it's like the ability to create complexity from simplicity is nearly impossible. So that's one of their big arguments on this. But
and I've heard that a lot, and every time I ask them, what is complex in this case, I can say that RNA itself is fairly complex. I can say the nitrogenous base that you use to create genetic material, I can say that's complex. I can say the fact that two carbons attached to one another in a single bond is complex. So where do we draw the line between complex and simple in this? Because if I'm in a laboratory, if I'm in my lab and I talk to my PI, my boss, and I go, this interaction looks complex, he's going to look at me crazy, because that doesn't mean anything. That it's a nebulous concept when you start talking about science, that you have to have some sort of metric by which you're going to label this. And that's fine. You can use that label. You can say simple, complex, super complex, but have some sort of metric to tell the difference between the two, or how many other categories you have. But for a younger creationist, you're not going to get that, because all they're really trying to do is cast aspersions here and there. And that's it. If they can cast aspersions, if they can just cast doubt on known science, then evidently they win. That's supposedly how it works.
Yeah, exactly. So if we look back on what we know about early life on the planet, going back to single cell organisms and stuff like that we have in the fossil record. Do they back up the idea of kind of an early kind of an idea, like a recipe for life that could be based off of abiogenesis?
Oh yeah, so if we're going to look in the past and sort of see what things were like and sort of the cocktails that were used, we can look in the present and sort of tell what happens. Because mind you, within our cells, we have, you could say, a foreign invader. Our mitochondria was not originally in there. The mitochondria that we have and that all of us have are actually a separate single-celled organism, completely separate, that at some point in time formed a mutualistic relationship with our early cell. And that acted in concert to create the life as we know it. Because now we can perform a different type of respiration that yields way more energy than fermentation, right? So that's something we can look at. We can also look at like early protists, for example, because a lot of single cell organisms that we have today have not changed very much from back then. Now, obviously, we can't really go back in time and tell what was what, right. But based on some of the conditions, we know the type of metabolism that they had. We actually have large formations of single-celled organisms that are near cliff faces and rocky shores. I think they're called stromatolites. I'm drawing a blank as to the name of them. But based on their structure, based on their location, based on certain geological samplings, we can sort of tell what occurred during that time period and have a pretty good idea as to the metabolism that these organisms had at the time.
Mm. Yeah, that's actually something I was going to touch a little bit, and I think you might have already described it pretty well. But there are environments on the Earth right now that are kind of similar to what we would have considered back when Earth it was just forming. Kind of when the oceans were just forming on the surface. Do we see any examples of abiogenesis occurring today, or is it because there's so much life, like you mentioned earlier, it's impossible to get that? I mean, there's got to be like some environment where we wouldn't expect life to exist that it does.
Yeah, that's a good question. So is there like some pool somewhere out there, some spawning pool, right, where life is just sort of generating? Not likely. Too much time has passed that life has proliferated everywhere, and that the life that, let's say you did find some, life form in a remote area, it would be more than likely an extremophile that's already existed for millions of years. That the only exception, and I just thought about this, there was a geological sample taken, and I believe it was Antarctica, where they actually did find an organism. It was a fairly large microbe, single cell, and this thing, I think they said, had to be several million years old, and it actually came back to life once they brought it up out the ice. So we can take samples from that and sort of see, well, what's the genome of this thing look like, and is it similar to what we have now? Because that will sort of validate or maybe invalidate what we've surmised based on some of the interaction of the anaerobic and the aerobic microbes that exist to this day, right? So a lot of that has to be sort of, I guess, deduced first, and then while we're sampling different things, we sort of see, okay, maybe we need to adjust our expectations here, become more precise now that more information is available about these organisms.
So, is there hope that that we can maybe find some evidence of this on other planets, like Mars, for instance? Is there any hope of that?
Oh, absolutely. I think the Curiosity rover. No, was it Curiosity? Maybe Curiosity. I may be confusing the names of the rovers. There's a recent rover that has found evidence of water on Mars, and that the presence of water is sort of a boon for anybody looking for life, because we really need it to exist. Every organism damn near needs water. But I will say that we pretty much have a good idea that any life that we find is going to be carbon based. And that's because the only real other option that we have as far as elements, and I'm not going to get too deep into the chemistry here, but the only other option really is not sulfur, I'm sorry.
Silicon based?
Silicon based life.
Yeah.
And the problem with that is that the energy required to break silicon bonds is enormous compared to carbon. Carbon bonds break if they're heated to a certain degree, but the heat needed to break a silicon bond is much more, I was just saying, not the heat, the energy needed is considerably higher. I think several degrees of magnitude higher. So the likelihood of us finding silicon-based life like in Star Trek, probably unlikely, very unlikely.
Interesting. So if some form of life, even if it's microbial, exists on other planets, is it possible to say that maybe some of that life came into Earth through like a meteorite or something like that to actually kind of kick off the process?
All right, I believe you're referring to panspermia, I believe it's called.
Yes, I believe so.
Yeah, that's a definite possibility. We know that there are elements out there that get sort of seeded on our planet from meteor strikes. And so if there are microbes, and let's say there are extremophiles, absolutely they can hitch a ride on some of these extraterrestrial objects and be seeded here on Earth and then begin to proliferate. That's absolutely possible. And so I think that's something that people should explore. And should we find life on some of these other planets or just indications of life, I think it gives more and more credence to that option.
Mm-hmm. So, I mean, that just basically says that abiogenesis isn't like the only thing that we can live on, but we'd have to at least find a way to explain how it exists anywhere, not just on Earth, but anywhere in the universe. There has to be at least that one point of creation and of life from something.
Right. And like I said earlier, it could be a multitude of factors that contributed to life as we know it on this Earth. And this is something, if you've never seen the new Cosmos with Neil deGrasse Tyson, he talks about this, that life actually started about 4 or 5 different times on this planet, and there were just mass extinctions, and the conditions became just unhospitable for life. And so that right there, just on our little terrestrial planet here, tells you that life is almost an inevitability if you have the right conditions. And when we find those conditions, such as like the Goldilocks planets that you hear people talk about, we sort of just expect life in some form. It may not look like us, it may not metabolize like us, but it will be very similar to us in that more than likely it'll be carbon based, just based on what we know about general chemistry.
Yeah, so going back to the idea of how it, a lot of creationists say it's so hard, it'd be like a one in, an outstanding number of possibility that life can actually be started through abiogenesis. Yet, like you said, we're finding that if life is possible, it tends to happen, or at least it did in our example here on Earth.
Right, and so they're making arguments from incredulity, essentially, and it's because I like to phrase it like this without bringing out the fallacy wiffle bat to beat them with immediately. I said, well, that's fine. You think about and if you take the span of time that our Earth was formed, and then you look at how many millions of years ago that we know life started to begin the first time, right? And you start seeing these different large chunks of time that grow closer and closer and smaller to the present day, you'll notice a kind of a bit of a pattern, you could say. Time isn't a factor. It's if you are given enough time you're going to have all types of possibilities become reality. And so if for 50 million years in our geological history, nothing occurred, well, that's cool. The Earth has been here for much longer than that. So if their argument is to say, well, I can't personally conceive of this happening, well, I personally can't conceive of 50 million years, and neither can most people, because that's way beyond our personal understanding. We know that millions of, well, if you're not a younger creationist, I guess, you know that millions of years have transpired, but you have to do a lot of wishful thinking to sort of, just sort of wish away facts about the world.
Exactly. So I guess the big question is, what are we doing now to advance the study of abiogenesis? So we kind of have these different theories going. Where do we take it from here?
So that would be a question better suited for somebody who's really, really big into early Earth chemistry. So I haven't really read too much since before COVID, but what I think the last thing that I read was that they're looking into to see how, and I should say, what variety of proteins could be produced, and would they have the correct conformation needed to perform enzymatic functions. And so let me sort of illustrate. You have a variety of amino acids, right? The basic building blocks of proteins. Enzymes are a type of protein. They need to be folded in a particular conformation in order for them to cleave, in order for them to attach other chemicals to other chemicals. And so that's a very arduous process if you don't have something doing the folding. Now we have organelles inside of our cells that properly fold proteins, because when proteins are not properly folded, you have prion diseases like mad cow disease, for instance. And so the question then becomes, well, how do you have a properly folded protein to do these things before you have other proteins to properly fold them? Well, what we do know is that, and there's I think it was Hans Christian. I'm blanking on the guy's name. The researcher many moons ago. He ended up taking some very basic proteins, and what he did was he denatured them. And so they're positive and negative charges, because each of these amino acids has its own charge. He eliminated those charges by putting them into a solution, so they lost their function, they lost their forms. And then he took them out of that solution, and they began folding back into their proper conformation, and they went back to doing the job that they were supposed to do, whether it was cleaving this or adding that or whatever. So that right there tells us that the information, if we're going to use that term, needed to fold them properly is within the amino acid sequence itself. So they'll sort of just innately fold into their correct conformations in most cases. Now, there are obviously exceptions to that. Something like, let's say, our hemoglobin, for instance, is a protein complex made of several different proteins. And so that needs a lot of assistance to properly get into position, and as well as something called a prosthetic group. But basic proteins already have what they need to get in their proper conformation.
So obviously at this point we'll be talking more about single-celled organisms, which would then obviously evolve into more complex organisms like what we would see today. Yeah. During this stage, we would only have, if we even got to that point yet, because these are just the basic building blocks of those single-celled organisms. So multicellular organisms wouldn't be something to occur until much later, because you have to have a way for these things to proliferate. Once you have the first one, it successfully proliferate at that, and then communicate with one another and act in concert to one another, as opposed to, just eating one another.
Yeah, exactly. And I guess that would be, is that a separate field of study right there? Because you go just from like chemistry into something a little more complicated at that point.
Yeah, that's a pretty specialized field. There's uh, endo symbiotic theory is sort of an interesting field itself. In that, because I mentioned this earlier, when you have our mitochondria, at some point in time, some eukaryotic cell that would eventually, create us, consumed another microorganism, but rather than breaking it down, it decided that, hey, for whatever reason, we want to use the energy that this thing produces, so we're not going to eat it. How that occurred, we have no idea. But somehow that occurred, and that when our cells replicate, it replicates. And then we have this mutualistic relationship with it. And that's, again, a very, very specialized field, because now we're millions of years after this event has happened. So how do you investigate that? So understanding the relationship between the mitochondria and the rest of the cell is really key. And I can't tell you how I would begin to even decipher that relationship. I have no idea. I think that gets into a lot of organelle kinetics and stuff, and cell signaling that is a bit beyond me. Now, mind you, I passed a cell signaling course, graduate level course recently, but I get way beyond my pay grade.
Yeah, I know I've seen material on that topic and everything, and it's complicated. So, but it's just getting to that point too. Right now, a lot of creationists are just saying we can't even get past the idea of creating a cell that can, as you say, come together, become alive, and then continue to live and breed and everything like that. So we're getting kind of ahead of ourselves to get into multi-cell from that point.
Right, and I've actually had this conversation, funny enough, with Otangelo, because he would understand, of all people, he wanted to understand, well, how do we even begin with these cells? I said, well, you begin with a micelle. And he didn't know what that was. I said, okay. I said, Otangelo, do you know how soap works? Well, what does this have to do with? I said, okay, that answers my question. So the way soap works is that you have these little hydrophobic particles. We'll call them that. They're compounds. We'll say particles. And because the water that we use to clean our hands with is polar, we're going to need something that's not polar to get like the dirt, right? Because if you have a polar substance with another polar substance, then they can mesh, but you have polar with non-polar, it's not going to work. So what ends up happening is that the soap that we use forms kind of a bubble almost. And when they form this bubble, they get to trap the non-polar substances within them. Now, you would say, well, Drag, I've seen cells, and cells are flat, right? They're not a bubble. Well, if you change the pH and the temperature, guess what happens? It flattens out. Guess what that looks like? Our cells. So it's that sort of interaction that we know that when you change these conditions, well, hey, okay, cool. The dynamics of these compounds can actually contribute to what we see nowadays. So figuring out the conditions for that is not too hard, because after I got to that part of the discussion with Otangelo, he's like, oh, you have to do that in the lab. And I said, oh, so are you telling me, Otangelo, that heat is only found in the lab, that a change in pH is only found in the lab, that pressure changes are only found in the lab? That's ridiculous. All of those are found in underwater seabeds, every last one of them. Mm. So it's, again, if you don't have the requisite knowledge and you don't actively go and seek this information, you're going to say dumb things like that. And so I think it's interesting when I talk to younger creationists and they haven't even researched, because you know what? Hey, no one knows everything, right? And I'm not going to browbeat anyone for not knowing these little science factoids, because it's not their area. But if you're going to tell other people, well, this can't happen, that can't happen, at least have a cursory understanding of basic chemistry, which I've yet to meet a younger creationist that could even survive one of the courses that I'm in charge of, much less a general chemistry course. So, right. They're just picking up lines from this website or that website, and it's Pratt, at the end of the day, points refuted 1,000 times.
Plus, with people like Atangelo, he's only taking information that fits with his own narrative that he's already gone in there with.
Oh, absolutely.
And so you don't know what he's throwing, what he's throwing out, what information he's looked at, and then gone, yeah, no, that doesn't fit with what my God says things have to be.
Right.
You know, which is just the wrong way about doing any sort of research.
Absolutely. Yeah, I don't just go and look at the stuff that I'm interested in. I have to look at stuff that supports sort of what my endeavor is, whether I enjoy it or not. There's lots of stuff that I would rather have not had to learn about the the orbitals of certain compounds and stuff, because I don't care. Like I don't use that at all ever. But everyone who takes general chemistry has to learn about orbitals. But that's what happens. You have, and if people are looking to get into these discussions with creationists, that's something to consider. They're tailoring all their information towards one conclusion. If you properly educate yourself, and it doesn't have to be a lot, just a month's worth of research, you will know far more than them in less than that month. I'm going to be honest with you. You can. It's not hard.
Yeah. Especially when they're coming in with such a bog-eyed view as, oh well, the Earth is only 6,000 years old. Which, just take any basic geology class or chemistry class, and at like elementary level, and you realise straight away that that's false. It can't possibly be true.
Yeah,
right.
And I mean, we're talking about just the basic study of what we experience here in nature versus the idea of waving your hand and things poofing into existence. It's not that hard to really come up with an argument against that. As we're getting closer to the second half of the show, I do want to remind everybody, if you have questions for Dragnaut, make sure to put them in the chat and direct them to the Borg Skeptic. That way we can make sure as we get to the end of the show, we can read them and hopefully get your answers.
Yeah. We actually have a question now if you want me to ask it, because I think it's on topic with what we were talking about. So
absolutely.
We have a question from Scott Williamson. Is it fair to say, because DNA has a unique double helix shape, like a twisted ladder, that this is a literal unique genetic code in all living organisms?
I guess you could say that. When you say that it's part of all organisms, then it stops being unique. But if you were saying in comparison to the shape of other, let's say, compounds, and sure, but it's just, it depends on how you depict that, because it can't be unique and ubiquitous at the same time. So I would just make sure that you relay that in a manner that you're contrasting it against something that doesn't already fit that description. Does that make sense?
Mm-hmm.
I don't know. I think that went over my head a little bit. So.
I hope that answered your question, Scott.
Yeah. If I didn't answer, I'll try to do better. But yeah, so there's let's say there's proteins are a perfect example of these basic substances sort of taking different forms, because one protein can look like just a ball of yarn, while another protein can look like the letter T. And so, their shape has a big influence on their usage, because there's certain points that which DNA does not take that double helix form, and actually when it needs to replicate, it will sort of turn itself into, you've seen a pile of cables mixed up. So it actually takes that shape at a certain point. So, and it'll change based on the phases of whatever the cell is actually doing at the time.
Well, Scott says, cool, that makes sense. So, yeah, he understood that.
That makes sense to you, Scott. All right. So, I guess if you're looking to kind of have this discussion with like a creationist or young Earth creationist or whatever, where do we start? If you're somebody like me that doesn't really understand the science, are there resources that we can use that can kind of help get this on a basic level where we can understand it? I know you said that a little bit of research goes a long way. Where can we start on that research?
Okay, so this is a fun topic because it depends where you want to go, and I'm trying to think of the best way to put this. Are you so? I tell people pick a lane. If you are more so into the philosophy, then cool. Focus on that. Stanford Encyclopedia of Philosophy is a great resource, but then understand science, the scientific arguments based upon Pratt. You don't have to go and take an undergraduate course in chemistry and biology. You can just honestly go to Crash Course Science on YouTube because there was some great, for just cursory information, great resources. But if you're trying to be sort of, I guess, or reside in my lane where I'm mostly science and then I have my philosophy sort of shored up to make great arguments, then I would start with AK Lectures on YouTube. I would definitely encourage people just to take some basic undergraduate courses in general chemistry 1 and 2, biology for science majors. And that'll give you sort of the basis for what you need to just understand what's going on, at least behind the scenes or under the hood. But you can't really get to the level where you can read scientific literature unless you take more advanced coursework. But if you're just sort of looking for a casual way to insert yourself, start with the Pratt arguments. And rather than doing what young earth creationists do, which is just sort of just specifically tailoring your knowledge towards the arguments, you understand what each and every concept entails. So if you're going to discuss evolution, then we're going to have to study a bit about genetics, specifically population genetics. In order to understand genetics, you're going to need that basis in biology, and you just sort of go back as far as you can go, the most broadest category you can get out of that, and then start from that point and work your way back towards the Pratt argument, because then that way you'll have an edge over people just offering basic Pratt, because I've heard a lot of it before, and I can tell you that people do well just addressing the Pratt arguments, but if they go off the script, people don't know what to say because they don't have that requisite knowledge. And so I would always suggest that people sort of go back, take your time, and just read some books. There's some great books out there by Raven. I think it's Biology, like 7th edition by Raven, John, and someone else. That is a wonderful resource. I actually have it in a box here somewhere.
Yeah, because this is a simple one. If you do just a search on abiogenesis on Google, it seems like actually more articles pop up for theists who are looking to argue against it. There's so many resources on why it's impossible and what to say to an atheist when they bring up the idea. So this is definitely something that theists are actually trying to weaponize right now and kind of put the arguments to rest in a way when brought up.
Right.
And especially for somebody like me who doesn't know how to argue about it, because I'm definitely not a scientist. You can quote a paper, and then they just quote a paper that's favorable to their side of the story.
We've actually got a good question in the chat which relates to this, so I'll get that asked now if you want me to. Yeah, absolutely. So it's from Scott Williamson again, and he's asking, why is the genetic code referred to as a universal code? A lot of creationists point to the universal code as an indication of a common creator God. How can we explain to them the real implication? Yeah, so it's called the universal code because you don't see an organism without DNA. That's why it's called universal. It's ubiquitous to each organism. We obviously exclude viruses because viruses are technically not organisms. But I've heard this argument before, and what they're doing is they're bootstrapping God into this. Because what I like to do is, that's fine. If you want to say that there's a universal creator, that's awesome. So how does the
creator actually play into this? And is there empirical evidence for it? Because if it's not, then we can't play, hey, I'm going to lean on science and evidence-less arguments, but have no empirical evidence to demonstrate it. Because what they're asserting is that there's some being, there's some entity outside of the laws of physics or whatever that is dipping its fingers or tendrils into reality and manipulating it. And I wouldn't know where to begin to even discern that that's happening, because I could insert the Borg. The Borg could be at the heart of it. I could insert chocolate cake. It's the same thing. And I think that people should not be afraid to just sort of go along with it and have them explain to you how they substantiate that claim, because it's absolutely ridiculous.
I'll just like to say, if you're putting the Borg into the argument, I'd be very skeptical about that.
There you go. Yeah, but I've heard this argument time and time again. It usually starts with an argument from incredulity and the insistence that this could have only have happened through a supernatural means, and then it goes back to my personal credulity. I don't think it. Okay, cool. I'm glad that you can cast aspersions. That doesn't substantiate your claim.
Yeah, I mean, you could, and correct me if I'm wrong on this. I mean, this would also easily just suggest that there was at some point there was a single organism that everything else came from. So it just kind of, as you're going back in time to an earlier form of basically what all life here is on Earth came from, you're going to find at least one point where we shared that common ancestor.
Absolutely. And arguments like that are kind of funny, because with the aspersions cast towards like evolution, you'll also get them cast towards common descent. Well, you can't argue against common descent, but then say that there's a common, designer or a common creator. Well, then that means that we descended from that common aspect, right? So you'll see people play both sides of that coin, but I've yet to actually have anyone provide positive evidence for any of this kind of stuff at all. And it's been years since I've been engaging in these discussions, and I'm still sort of waiting on who wants to insist that there is empirical evidence for a supernatural entity, because that would blow my mind how one was able to do that.
Yeah.
And they'd have to, if they did have that, they'd have to go and step up for a Nobel Prize, because.
Oh yeah, absolutely.
Because like we've been saying, it's like they're just adding it into the equation. It's not necessary, because we can give a natural conclusion for all this stuff.
Right.
They simply are just putting in an extra step that is not necessary.
One fun thing to ask creationists too, since a lot of them like to pretend to play scientists, what would invalidate their hypothesis? That's a great question to ask them. So what would need to occur? What evidence could you find that would preclude their God hypothesis in this manner?
Mm-hmm.
And none of them have even thought about it. And I said, well, if we're being scientists, we're talking shop, right? We're all sitting around the lab. Then let's do that. Let's be rigorous about this and understand that, because if I'm in my lab and I say, you know what, there's the FET2 gene in yeast, is the source of a selective rescue mechanism during cell aging, right? My scenario that would invalidate that is that if I inactivate FET2 and the cell gets rescued anyway, right? So I can tell you just here, and that actually is close to what I'm actually studying, but that right there gives a very simple explanation as to how that works and how science works. But for creationists, they don't care. They'll just posit things and insist that it's right because, well, they've already got their conclusion.
Mm-hmm. All right, we're definitely getting closer to the hour. Do we
don't actually have any questions in the chat, but at the moment. But I was wanting to, because we've covered this topic pretty well here, Drag. I was wanting to ask you about the research that you're looking into at the moment within your thesis, the cell aging. Is there anything interesting that you could tell us about what you're doing there?
Yeah, so that's actually taken a back seat to the Cure Lab, which is the undergraduate research experience. But essentially, what we see happen during cell aging, and this is in our cells and in yeast cells, because my model organism that I use are yeast. Uh, there is an alkalinization that occurs within the cell. So as the cell gets older and it replicates and replicates and splits off and all that good stuff, we see it become more and more basic. And that we believe is sort of a part of a systemic failure at some point, because there is a protein complex called an ATP synthase, and all it does is pump hydrogens into the cell, into a specific space in the cell, I should say. And if that particular protein complex is damaged through aging, through whatever, then it's not going to efficiently pump those hydrogens into that space. So it's going to become more and more basic over time. So what I do is that I genetically damage these protein structures, these complexes, ATP synthase, so that it mimics the cell aging. And so then we try to add in different sort of reagents to see, okay, if I add in this much salt, or if I add in paraquat or ferric chloride, what happens when the cell takes up these substances? Will they survive? Will they continue to grow? Will they grow better? And so we need to see, sort of what cocktail best creates the environment that this cell needs in order to obtain enough survival that we can say, well, it's significantly different from the wild type that we have here, which is a separate, cell strain. So what that basically looks like is that I have it's a metal device with a bunch. It's like a plunger, and it has a bunch of little metal forks on it. And what I can do is I could take a 90-well-6 plate, like a tray, and I can put in different substances that I want to test. And I have a strain or 6 of different yeast strains, some that are damaged with that ATP synthase I told you about. Right. One that's fine, it's a wild type, that's just my control, and a bunch of other different ones that have different metabolic things activated or deactivated. And I can test all of those substances with all of these different strains, and then grow them on a plate, and then see, how they grow at different concentrations. And so something like that will take me a few days to do. And I take pictures of it, and we sit down and I talk to my PI, and we try to surmise what's going on. There's a bit of quantitative data that goes along with that, where we put it in a plate reader for about, a day, if we're doing it just inside that tray. And so we can see the density change based on the growth. So the more dense it becomes, the more growth that we're seeing with this particular substance, whether it be the salt or the paraquat or whatever. I know that's a lot of stuff, but that's I hope that I didn't get too deep into that. Y'all kind of picked up the gist of it.
Yeah, of course. But extremely interesting stuff. It's something I think we could probably do another show on that topic alone.
I agree.
Definitely. We are actually getting a couple of questions in the chat now, so we are coming up to the end of the show, so I'll get these asked. 2 sets of in the wrong screen. There we go. Yeah. So, question from Joshua. What is the role of autocatalytic processes and their role in development of the life process?
Okay, so it's kind of a chicken and egg thing. I sort of explained this earlier. It's when you have the processes now to create life, we have helper enzymes to facilitate that process. But back when there was no life, you didn't have that. So how did we go from this substance here to substances that are way more complex and that would eventually lead to life? So one of the ideas is that RNA specifically could catalyze its own reactions that would eventually lead to those proteins that would then later help facilitate a more efficient process. So let's say you have a factory and the factory doesn't actually have any helpers. The guy, sort of does it by himself, but then he does it by himself. He creates the widget, and then this widget then goes out. He can make money and hire more people, right? So we're at this stage now where there's a bunch of people in the factory creating widgets, if that's a good enough analogy.
Yeah,
I think so.
I think Joshua will be happy with that answer. We also have another question from Theoretically. Where does drag a line, considering ethical limits of stem cell or animal experiment research, GMO, cloning, etc.?
That is an ongoing topic, believe it or not. And all of my graduate courses, we sort of spend a little time sort of discussing that, even undergrad. So I think here in Texas, embryonic stem cell research, you can't have a viable line, extend past 9 weeks, I think it is. And I'm not sure why the line is drawn at 9 weeks. I honestly couldn't tell you. I'm not in biotech, but I'm fine with that. I'm fine with, us using embryonic stem cells to sort of see what happens, because that's such fertile ground to do research in. I have no issue doing that. Now, if we were doing something that I consider unethical, such as, harvesting from people that, like, without their consent, obviously that's a big no-no for me. Consent has to be 100%, involved in this. So if there is human waste materials from like a spontaneous abortion or something like that, then I'm all for using those, because they're going to be incinerated anyway. I'm all for using that for biotech purposes. And I think there is cloning. So I am actually a proponent of cloning, but for animals, not for humans. And that it should be for obvious reasons. But I think we start crossing a lot of ethical lines. And we start, if anyone's seen The Island, the movie The Island, that's sort of the territory that I never want to go in. And I think that, we learn a lot through cloning, but it should be kept fairly limited and within the range of whatever the IRB sets forth. And IRB is pretty much a group of people in a facility, in a lab, that monitors and makes sure that people are doing what they're supposed to be doing. And they actually make guidelines as far as who can do what.
Theo says thank you for the answer. So great answer there. So we are coming up to the end of the show now. We're coming up to the hour. Is there anything else that you wanted to ask, Skids?
No, other than where can people find you there, Dragnot?
Oh, I do run a YouTube channel. It's about drama. I wouldn't watch it. But if you want to see me on, talk, I do occasionally do stream from my lab. The problem is that the reception's so bad in this old building. I can't wait till we move. Is that it cuts out a lot. And so as I'm explaining stuff, I can't really tell when the signal goes out, and if I need to go to the back of the lab and show a whiteboard because I had to illustrate a chemical reaction. The connection just shit. So what I might start doing is maybe pre-recording certain things and or talking to the IT guys to see if I can get access to the Wi-Fi and stream, because they've found a way to not allow people to stream or even be on Discord on Wi-Fi. Because they're dicks, I guess. But yeah, I am on AXP. I'm on Talk Heathen and The Line and Hang Up and all those shows that people love. If y'all like me on there, just email and harass Matt, and he'll call me and I'll come on. That's literally how it works.
That's what we need to do. So got our homework ahead of for the week. Yeah, I would love to see streaming from like your lab and some of the work. That would be awesome if you can make that happen.
We actually Titans just put your channel in the chat for anybody that is interested, and I think we also have the I know we do. We have the channel link in the description of this video as well. So I'd just like to say thank you very much, Dragnot, for coming on. It's been an amazing conversation. Learned a lot, and we've had quite a numerous amount of viewers watching and learning from what you've had to say. So thank you very much, and I can imagine thank you from the people in the chat as well.
Cool, it's my pleasure.
I think at the beginning, Skids did mention that we have other shows on the channel, so you can find us here every Wednesday at 8 p.m. I think is the new time that we're setting. You can also find Bryce and Caleb over on Secular Soapbox on a Monday at 8 p.m. And you can find me and whoever is my guest host for the week on the Bo Skeptic Show on a Tuesday, which is at 8 p.m. again.
Don't forget Michael Weissman on The Bible Says What?
Definitely, yeah. That's a pre-record show that we usually do. So check that out at 8 p.m. on a Thursday.
Absolutely.
And once again, thank you to all of our subscribers for helping us get to the past our first milestone of 100 subs.
So excited.
Congrats.
Thank you.
Yeah,
Thank you.
Thank you very much. And as always, stay home, stay safe, and wear a fucking mask. Thank you and good night.
See you.